Welcome back, students. We're going to begin Unit 11 with a study of the DNA molecule. You can find relevant reading for this unit in Chapter 10 of your biology book, specifically sections 10.1 to 10.3. First, you've probably heard of the DNA molecule. We all have. And we know that it's the molecule responsible for inheritance, that is, the traits that get passed on from one generation to another. But for a long time, scientists, well, they knew that something got passed from parents to offspring, but they didn't know exactly what it was. For centuries, scientists knew that there were traits passed from parents to offspring. We had used the term genes for a long time before we even knew what DNA was. Whatever that unit that got passed from parents to offspring, we gave the term genes to. And that term is still valid today. It's just that now we can tie a gene to a specific molecule. And that happened relatively recently. In the broad scope. When I say the 20th century, specifically, we're talking about the year 1953 when the scientist Watson and his friend Crick published their theory of how DNA was responsible for inheritance. And it all ties into the structure of that molecule, which is what we're going to take a look at today. So you may say, geez, 1953 was a long time ago, but in the broad scope of things, we've known that inheritance was going on for many, many centuries, and only in the last 80 years or so did we come up with an explanation for it. One of the key experiments that led us to the discovery of DNA as being the molecule responsible for inheritance was actually performed in the 1920s. Robert Griffith was a famous scientist, and the gist of his really, really important study goes like this. He was doing experiments on animals, specifically mice, and he was injecting strains of viruses into them. He gave them names. So let's say there's an R strain of a virus that was injected into a mouse, and the results are the mouse stays healthy. Let's take the same type of mouse and inject a different strain of virus in, the S strain, and all the mice die. Then let's take the S strain bacteria or virus and heat them. Heat them to a point of boiling where the protein or DNA or whatever the molecule of inheritance was would not be effective anymore. Inject the heat killed S strain into the mouse, and the mouse survives. Then the final and most interesting experiment that Griffith did was he said, okay, let's take a living R strain and a heat killed S strain, both of which had produced healthy mice up above, inject that into the mouse, and aha, the mouse dies. Something, some substance must have been transferred from the living R strain to the heat killed S strain, or perhaps from the heat killed S strain to the living R strain. And in that case, it transformed the R strain into something deadly, and the mouse dies. Well, guess what, students? Whatever was in the heat killed S strain and was transformed into the R strain was found to be DNA. At the time in the 1920s, this was really, really surprising because DNA, although it was a molecule known to exist in the nucleus, was not a complex and still is not a very complex molecule. There are many, many, many more types of proteins in our body than there are types of DNA. We know of 23 different chromosomes or types of DNA in our body, but there's thousands upon thousands of proteins. And scientists and Other people figured, hey, the traits that get passed on from parents to offspring are awfully complex. So wouldn't it stand to reason that a complex molecule like a protein would be responsible for it? Well, as the work of Watson and Crick in the 1950s showed us, it doesn't have to be a complex molecule in order to be responsible for a very wide variety of traits getting passed on. There's an elegant simplicity to the DNA molecule that we have the privilege of studying now with the benefit of centuries and of good reason and good scientists who laid the foundation for us. So let's dive in. The DNA molecule stands for, you may have heard this acronym, deoxyribonucleic acid. Think back to our unit on nutrition. There are 4 types of organic molecules: carbohydrates, lipids or fats, and proteins, all of which we studied in the nutrition unit. And then there was a 4th one that we said, we're going to set this aside now and come back to it later. Well, that 4th category of organic molecule was nucleic acids. And here we are coming back to that. We're indeed looping back to the DNA. So DNA, the molecule that codes for and directs the development of nearly all organisms on Earth. A nucleotide is a nucleic acid, or a nucleotide is a subunit within a nucleic acid. If you take a look at the picture on the right, each of these units where you've got a dark green double hexagon and then a light green pentagon and then a yellowish circle, that's a nucleotide. We'll take a look at that in a minute. Here is another nucleotide, and here's a 3rd nucleotide. So this is a chain of 3 nucleotides, each of which consists of a phosphate group, that's the yellow circle, a sugar, in this case it's ribose sugar, that's the ribo in deoxyribo, and a nitrogen-containing base, that's the either double-sided or a double hexagon or a single hexagon shown on the right. DNA is found in the nucleus of eukaryotic cells, or as you might recall, prokaryotic cells don't have a nucleus, and so they're just jumbled up in the cytoplasm of the prokaryotes. Okay, let's take a little bit closer look at an individual nucleotide. Remember the phosphate ion from our study of chemistry, PO4 with a 3- charge? Students, take a look here. Phosphate, there's a phosphorus atom, and then 1, 2, 3, 4 oxygen atoms. That's a phosphate group. Our sugar here happens to be called ribose sugar, but it has 1 fewer oxygen atom than a typical molecule of ribose, hence deoxyribose. And then the nitrogen-containing base, look at all the nitrogen atoms within it. Do you need to memorize this structure? No, not at all. But I do want you to know that a nucleotide contains a sugar, a nitrogenous base, and a phosphate group. So that's the key points in our notes here. Each nucleotide has a phosphate, a sugar, and a nitrogen-containing base. Now, let's take a look a little more closely at those nitrogen-containing bases. They have funny names, and yes, by the end of this unit, you're going to have to know at least the names of those nitrogen-containing bases. Okay, I'm going to reveal the names now. Adenine, cytosine, guanine, and thymine. In DNA, those 4 bases are what makes the differences between the really, really long chains of nucleotides. In the previous slide, we saw a chain of just 3 nucleotides. Well, in the DNA in your nucleus, there are hundreds of millions of nitrogen-containing bases. And those are what join the 2 segments of DNA together. We abbreviate them A for adenine, C for cytosine, G for guanine, and T for thymine. A, C, G, and T, just the first letters of their names. Okay, now I know you're going to freak out with this. Students, this is just the full picture of the 4 nucleotides of DNA. You're not responsible for knowing this, but just take a quick glance here. Every single one of these nucleotides contains a phosphate, a sugar, and a nitrogen-containing base. Phosphate, sugar, nitrogen-containing base. Phosphate, sugar, nitrogen-containing base. Phosphate, sugar, nitrogen-containing base. The only difference is the type of base that's within the DNA. Okay, so let's get back to that idea of a simple molecule coding for huge amounts of genetic variation. Okay, little bit of math. Statistics question. This is a permutation. How many different ways can you arrange 3 different, 4 nucleotides in 3 positions in a 3-position sequence? So if your only choices are adenine, cytosine, guanine, or thymine, that's 4 choices, and you can have 3 of them in succession, how many different arrangements of A's, G's, C's, and T's can you get? And it's okay to repeat. You could have, for instance, adenine, 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 or you could have thymine, thymine, thymine. How many different letter combinations could there be? I'm going to pause the video. You go ahead and pause that video for a second. Think about it, and then come back. You probably stumbled upon this, students. It takes a 4 raised to the 3rd power to figure out all the possible letter combinations. In other words, you have 4 possible nucleotides in position 1 of the sequence. You have another 4 nucleotides in position 2, and another 4 nucleotides that could be in position 3. So 4 times 4 times another 4 is 4 raised to the 3rd power, which works out to be 64. There are 64 ways to arrange just 3 consecutive nucleotides. Well, consider this. Every piece of DNA in our genome, that is the sum total of DNA in our nucleus, is millions of nucleotides long. Millions. And so what that means is, even though DNA is a fairly simple molecule by structure, its really long length allows for a very, very high degree of genetic variation. Okay, here's a picture of 2 strands of DNA interacting with each other. There was a really important study done once we figured out that DNA was indeed looking to be an important molecule for inheritance. And Watson and Crick used this information to come up with their model of DNA. It was found that the amount of adenine and thymine in any organism is always the same. There are the same number of adenine molecules as thymine, and there are the same number of cytosine molecules as guanine molecules. And that always that was puzzling. That was puzzling. Scientists didn't know why. Why would there always be the same amount of adenine as thymine and the same amount of cytosine as guanine? Well, turns out these nucleotides are complementary to each other. That is, the adenine always binds with thymine in DNA, and cytosine always binds with guanine in DNA. They're pairs. They're always going to be pairs. And that led us to the big picture. DNA is a double-stranded twisting molecule. Double-stranded, meaning there's always going to be 2 pieces of DNA wound up around each other. And twisted because they're paired together, and it just so happens that that causes a twisting or coiling of the molecule. The backbone of the molecules, the strands of DNA, are the bonds between sugars and phosphates. The strands of DNA are anti-parallel. And what anti-parallel means is that one strand is opposite in orientation of the other strand. So for instance, let's trace the strand that's on the right side at the top where there's a free phosphate group. If you follow it along all the way to the bottom, it ends up with a free sugar group on the bottom of the molecule. The other strand, where there's a free sugar on one strand, there's a free phosphate on the other. Let's trace that strand all the way up. Aha, that's the one that has the free sugar group at the top of the molecule. If you were to zoom in on the connections between the two strands, you would see the nucleotide bases, the nitrogen bases, thymines and adenines, cytosines and guanines. They all pair together, A's with T's, C's with G's. And the specific order of those bases is the genetic information encoded in DNA. The first scientists to put this together and actually to publish it were named Watson and Crick, and their discovery was to much fanfare, and there was quite a bit of politics behind it. They may have indeed stolen scientific information from some of their colleagues, and no good discovery is without its own story. There is a fantastic video on the journey of Watson and Crick and some of the people they left in the dust without mentioning that you could take a look at on Netflix. But that's not really the focus of this course. It's not the history of science in itself, although it's kind of interesting. A couple other people we really should be mentioning. If we're mentioning Watson and Crick, we need to mention Rosalind Franklin. Dr. Rosalind Franklin was the one who produced the X-ray crystallography images that gave Watson and Crick the idea that, hey, there's something in the nature of DNA that makes it a twisted molecule. And it was Dr. Franklin's image shown here on the left that Watson and Crick, I guess, stole and used to make their own synthesis of the information. We should also mention there was a fantastic Sister Miriam Stinson, who worked at the University of Michigan, and she prepared a method of making DNA bases analyzable using potassium bromide. And it was that preparation method that allowed Dr. Franklin, who was working in Paris, to capture the images, and then Watson and Crick, who were working in London, stole Dr. Franklin's images. So there's a long line of credits behind the discovery of DNA. Okay, now to the actual biological molecule. DNA is an extremely long chain of connected strands of DNA that are twisted upon each other. If you can envision coils upon coils upon coils getting packed really tightly into a compacted structure called a chromosome, that's what you find in the nucleus, are coiled up, packed tight DNA, where there's 23 types, and each DNA, each cell in our body has 2 pairs of each type of chromosome. And we'll get into that when we study genetics. Okay, here's a what they call a DNA karyotype. The 23 different types of DNA in the genome, all 23 types of DNA together are called your genome, and we are now able to sequence your genome. In other words, we can take a sample of DNA from any of your cells, run it through a machine, and with the aid of some really powerful computers, come up with the A's, the T's, the C's, and the G's that make up your DNA. Every type of DNA has genes. Now, not every single portion of the DNA molecule codes for a protein, as we'll see in the next couple of chapters. But every gene has a specific location on one of the 23 types of DNA. You might wonder, why are these strands of DNA banded? Why are they? What's with the stripes here? Well, each stripe is showing conveying the idea that there are lots of different genes on every single type of chromosome. In fact, there are hundreds, if not thousands, of genes on every single piece of DNA. Okay, I mentioned earlier that DNA is packed tightly in the nucleus. Well, there are other proteins that help to pack the DNA tightly. Think of this, students. Let's say you have a really, really long strand of wire, maybe 100 feet long, okay? And you want it to not get all tangled up. Well, you could use a spool. A spool helps you to wrap a really long strand around a central place so that it stays organized and doesn't get all twisted together. Well, the spool for DNA is a histone protein. That's what helps DNA package really tightly in the nucleus. And as we'll see soon, the process of cell division makes sure that the DNA gets packed tight into chromosomes before dividing so that you can do so in an organized way. Here's a nice image. We've got two strands of DNA. Sometimes it's described as a ladder that's twisting. So the rungs of the ladder are the nitrogen containing base pairs, and the rails of the ladder are the sugars and phosphates that make up the nucleotide. So you've got a really long strand of DNA, double-stranded DNA molecule, twisting and twisting and twisting, and the histone proteins are like the spool around which the DNA wraps and wraps and wraps and gets really, really packed tight in the nucleus. Okay, just a little bit that will help us have some terminology for when we study replication. When DNA replicates, that is, makes a copy, it forms a duplicate copy of every chromosome. Those duplicate copies are called sister chromatids. So take DNA type 5. All right, it's one of your chromosomes. You have, of DNA type 5, a version inherited from your mother and a version of chromosome type 5 inherited from your father. Separately from having two versions, you can also duplicate the DNA so that you have two copies of your mother's version of chromosome number 5, and you would have two copies of your father's version of chromosome number 5. The copies are called sister chromatids. Sister chromatids attach to each other at a site on the chromosome called a centromere. Here's a nice picture of a centromere. So one of the strands of DNA is called a chromatid, and if you put two of them together, we call them sister chromatids. The region where they attach to each other is called the centromere. And just one more term, the end or tip of the chromosome is called a telomere. Telomere, the end things. By the way, telomeres may be involved in the aging process. It's an area of active research. So let's say we've got lots of types of chromosomes stained and prepared on a slide under the microscope. And you say to yourself, okay, look, there are some short, stubby chromosomes and some really long ones. That's right. Different types of chromosomes contain different amounts of genetic information, and thus they are of different lengths. That doesn't mean any one, even the short chromosomes, don't have lots of information packed in them. It's just that some have more information than others. So, in any case, the region where two sister chromatids attach to each other is called the centromere region. And that's where we'll end our notes for today.